Bioinformatics Micromodules. What is sequence alignment? In this module, we will talk about the meaning of sequence alignment and why is it important in bioinformatics. Many types of biological objects can be represented as sequences, which are sequential chains of monomers. For example, DNA sequences are commonly represented as a string, where every character comes from the alphabet A, C, G, and T, corresponding to the 4 types of nucleotides. Similarly, RNA sequences are also commonly represented as a string with 4 possible types of characters. And for protein sequences, the characters come from an alphabet with 20 possibilities, corresponding to the 20 amino acids. There are other types of biological objects that can be represented as sequences too. When we study sequences, one important concept is the evolution of sequences due to mutations. For example, suppose that in the past, at the time that we call time t0, there are 2 copies of a sequence, which we call R and S. They are identical at that time. As time went by, some mutations happened. For example, some of the nucleotides were being replaced by some other nucleotides. There were some insertions, and there were some deletions. So after a lot of time, when it comes to the current time, which we call T, you'll observe 2 sequences that are similar, but not exactly the same anymore. This is actually what we can observe right now. In reality, we'll never be able to observe the 2 sequences at time t0. So one thing that we want to know is the correspondence of the nucleotides in the final 2 sequences. So we want to deduce their correspondence based on the current sequences. Alternatively, if we are not sure whether the 2 sequences actually came from the same origin, we could also try to find the correspondence and compute a similarity score accordingly. The idea is that if the similarity score is high, the chance that the 2 sequences actually came from the same origin is also high. There are many situations that we want to find out the correspondence and to compute the similarities. For example, suppose that we are studying humans and chimpanzees, and we want to know how similar their genomes are. The term genome means that the whole set of DNA from the different chromosomes. For example, for humans, the haploid set of genomes has around 3 billion characters. Computationally, when we want to study these kind of questions, we are essentially comparing 2 sequences, and we want to compute a similarity score between them. In another situation, suppose that we have found a gene that causes obesity in mice. Of course, we are interested to know whether this gene is also present in the human genome. So how do we find that out? Essentially, we can also represent this as a computational problem, where we have a sequence R, which is the mouse gene, and we have a database D of sequences, which are the human genes. For each sequence in the database, we want to compare that with the mouse gene and to compute a similarity score. So for any gene in the database with a score higher than a certain threshold, we say that this is possibly a homolog of the mouse gene. In the third situation, suppose that we are studying a certain disease called sickle cell anemia. Let's say we have already found a gene that is related to this disease, but we are not sure exactly which mutations cause this disease. So what do? We collect the sequences of 100 patients for this gene, and correspondingly, the sequences for 100 normal people. We want to find out the disease-causing mutations. How do we do that? We can formulate that as a computational question too. Basically, we want to find out the positions in the sequences that can separate the 2 groups of sequences well. To be more concrete, let me give you an example. Let's say we have collected 3 sequences from the patients and 3 sequences from the control people for that gene. As you can see, the different sequences are not of the same length. due to mutations happen at a certain time in the past. So in order to find out positions that can distinguish the two groups of sequences well, we have to first add some gaps to the sequences so that the characters in the same position correspond to the same character in the original sequence at time t0. We call this step alignment. After doing that, if we now focus on position 5, then we see a very clear signal that all the patients have a character G there, while all the control people have a character T there, which strongly indicates that that is related to this disease. Based on this information, we can then do more experimental validation to see whether this is really the causing mutation. So as you can see from this example, adding the gaps, that is, doing the alignment, is an important part when we want to study the disease-causing mutations. And similarly, if you want to compute similarity scores between different sequences, this is also a very useful step. So now let's formally define the meaning of sequence alignment. Suppose you are given a set of sequences. They can be of different lengths. You want to insert gaps into these sequences with two requirements. First, after adding the gaps, they have to all have the same length. Second, we don't want to have one single position having only gaps in all sequences, because that's not meaningful. Let's see an example. Suppose we have these two sequences. Again, we can see that they do not have the same length. There are many different ways that we can align these two sequences. One way is to add a gap between the two Cs in the second sequence. Let's check whether this is a valid alignment or not. We see that after adding the gap, the two sequences have the same length, so requirement number 1 is satisfied. Also, we cannot find any single position containing only gap character in the two sequences, so requirement number 2 is also satisfied. There are some terms that we commonly use when we talk about alignments. If we look at the sequences and we find the two sequences have the same character, we call that a match. If they have different characters, we call that a mismatch. If one of them contains a gap, we call that an indel. Indel stands for insertion and deletion. The reason that we call it this way is because evolutionarily, we are not sure whether that is due to an insertion to the first sequence or a deletion to the second sequence. There are many different ways to do the alignment. So how do we determine whether an alignment is good or not? In order to answer this question, we need to make an assumption that mutations are rare. If you compare two different alignments, one with fewer mutations than the other one, we'll believe that the one with fewer mutations is more likely to be the correct alignment. So we are going to define similarity scores of the two sequences based on alignments with few substitutions and indels. There are many different variations of the sequence alignment problem. For example, depending on the number of sequences, if you have only two sequences, that is called pairwise sequence alignment. If you have more than two sequences, it is called multiple sequence alignment. Another aspect is which part of the sequences do you want to align. If you want to align the whole sequences, it's called global alignment. Sometimes the sequences are only similar in the middle part. For example, in here, we're showing the alignment of only positions 2 and 3 of the two sequences. If we do that, this is called a local alignment. Finally, there are different ways that we can define the substitution scores, and we can handle the gap in different ways. To give you some concrete idea of alignment, let's consider a very simple setting. Here, we consider only two sequences. That is, we are doing pairwise sequence alignment. We're going to align the whole sequences. So this is essentially global alignment. And for the alignment score, we're going to use a very simple scheme, where for each position in the final alignment, if there's a match, we're going to increase the score by 1. Otherwise, that means either mismatch or indel, we're going to deduct the score by 1. So if you have two sequences, ACG and AGG, let's consider two different alignments. The first one is to insert a gap between C and G in the first sequence and after the second G in the second one. So here we have one match, one mismatch, and two indels. So the alignment score will be 1 minus 3, that is minus 2. Another possible alignment is not to insert any gaps, because the two sequences already have the same length. In this case, the alignment score will be 1, because there are two matches and one mismatch. Finally, let me show you a real example. This is an alignment that I took from a database called PFAM, protein family. So this is from the family PF00001, which is for 7' transmembrane receptor of the rhodopsin family. I'm showing only part of the alignment. There are more positions and more sequences. But from this part of the alignment, we can already see some interesting things. For example, if you look at the second position, all the sequences have the same character there. That means this position is highly conserved. On the other hand, if you look at another position, the different sequences actually have many different types of amino acid. We call that a relatively lowly conserved region. So when you look at an alignment, you can check the different positions to see their degree of conservation.